Hey, what's up, everybody? Welcome back to our next video in the STEM series of Pathways Education. My name is Max,
And I'm Kishan.
and we're excited to teach you guys about this special topic on protein synthesis. And so, for protein synthesis, some of the foundations for how a protein made is through transcription and translation. And as you can see in this diagram, we notice that DNA is in the nucleus and it ends up getting transcribed into RNA, which then ends up going out of the nucleus, attaching with the ribosome. And as you can see, it is translated into a polypeptide, which then attaches to an effector molecule and becomes an active protein. And so this is the generalized process for the production from DNA to RNA and then to a protein that can be utilized within the body. And so now we're going to kind of go more in depth with how proteins are specifically made, and we're going to start with the basis of it, which is DNA replication. And so just a little bit of a concept check. We want to ask you guys, what does DNA stand for and where is it found in the cell? And so we'll give you guys just a few seconds to think about that, and we'll move on. So for our little concept check answer, DNA stands for deoxyribose nucleic acid. And where it's found is it's found in the nucleus of every single cell in your body. This is kind of like, you can think about the nucleus as your information system. And so back again to that initial picture I showed you guys, here's the big picture, right? DNA is found within the nucleus. As you can see, the nucleus is this like blue circle within the cell, and then the cell itself is encompassed by this like orange color. This is where the protein is actually made within the cell. And so on to another concept check, kind of going into more on how the DNA like base pairs with each other. What four bases is DNA made of, and how do these base pairs pair with each other? So we'll give you another 10 seconds to think about it. And now for the answer. What we have here is that your DNA is composed of these four base pairs: adenine, thymine, guanine, and cytosine. And the way they fit together is that adenine pairs with thymine, guanine pairs with cytosine. And so what you can think about is they're kind of like puzzle pieces. Adenine is only able to fit with thymine, and guanine is only able to fit with cytosine. And if you were to try to fit guanine with thymine or adenine with cytosine, those puzzle pieces would not be able to fit together. And so now for a little bit of a review on DNA replication. The basis for how this works is that you have your DNA in the nucleus, and it ultimately unravels and unwinds. Then your top strand and your bottom strand of DNA end up separating, and you have this protein, DNA polymerase, that comes in and attaches to the site of where it is unwound and separated. And pretty much what happens is, for each of the top strand and bottom strand, DNA polymerase helps create a new matching strand for each of these top strand and bottom strand. And once the replication occurs, DNA will just ultimately twist back together into its double helix structure.
And so now we're going to move on to our next topic or subtopic for today's presentation. And so we're going to talk a little bit about proteins, and then we'll move on to talking about how they're made via transcription and translation. So a little bit of a basic introduction to protein synthesis. Proteins are long molecules made up of smaller molecules called amino acids. So you guys can think of amino acids as the building blocks of protein. So we can put together a combination of any of the 20 essential and non-essential amino acids together to put together a polypeptide, which is called a protein. And so we have two types of amino acids, as I stated, essential and non-essential. And our essential proteins are obtained through our diet. So that's what we eat. Foods we eat throughout the day contain certain amino acids that our body needs on a daily basis. And then we have the non-essential amino acids that are just produced in our body. So that means cellular processes in our body are creating these amino acids, so we don't need to consume them. They're being made in our body. And so the bottom left shows a basic structure of an amino acid. So on the left-hand side, we have the amino group, which contains a nitrogen. On the right-hand side, we have the carboxyl group, which contains a carbon, an alcohol group, and an oxygen group. And then we have a side chain, which is called the R group, which is variable. So that changes in between different amino acids. So for example, alanine and cysteine would have different R groups, and that's what makes these amino acids unique. And on the bottom right-hand side, we can see that we've grouped these 20 amino acids into different sections based on their individual properties. And so you guys can take a little bit of a look at that. It's a little bit complex and in depth, but I think it gives you a good idea of what these amino acids are and how they can be sorted and grouped. So now, what are proteins? Proteins are a basic unit in our body that we would not be able to live without. They serve some of the most important functions in our body. Some of these functions are to speed up essential chemical reactions in the body. So the enzymes in our body or in the catalysts that help propel the reactions that are going on every second in your body are proteins. And they look like the image on the top right-hand side. They're required for the structure of some of the body's vital organs. They help with recovering from an injury. Then they also tell your body when to grow and how much to grow. And they also provide cellular structure. So without proteins, as you guys can see, a lot of the cellular processes in our body and the bodily mechanisms would not be able to function properly. And two examples of proteins that I think are very applicable in our day-to-day life are keratin and hemoglobin. So keratin is the protein that makes up your hair. So if you guys have ever seen in a basic convenience store or a Rite Aid or a CVS or Walgreens, sometimes a lot of companies make keratin-rich products, which help with hair growth and keeping your hair nice and strong and silky. And also, there's hemoglobin in our body. And hemoglobin is the vital protein in our red blood cells that carries oxygen. So if it weren't for hemoglobin, we wouldn't be able to transfer oxygen from our lungs, which we breathe in through our mouth, to different parts of our body. And therefore, we would die if we did not have hemoglobin. So they serve very important functions in our body. So a quick question for you guys. Feel free to pause the video. So how does your body make these proteins? I kind of hinted at it in a previous slide, but try to recall and think about what processes in our bodies help make these proteins. We'll give you guys about 10 seconds. All right, and another question to think about. How does DNA code for these proteins? So how are we able to get from the DNA that we saw when Max was presenting in the nucleus all the way to a protein, which we see in the bottom right on this slide? How are we able to get from that genetic code to a molecule like a protein, which is so important for our body to use? So I'll give you guys about another 10 seconds to think about that, and then we'll move on. So this all happens during protein synthesis. which is a conversion from DNA to RNA to protein. And that is the central dogma of all of science. And that's how all living creatures function. In order for the body to make proteins, the mRNA, as Max was saying earlier, must travel outside the nucleus to the ribosome, which is also made up of some RNA and some protein components. And this step is called transcription. At the ribosome, a protein is formed from a chain of the amino acids, which we discussed earlier, which are the building blocks of these proteins. This step is called translation. And together, transcription and translation make up protein synthesis. So DNA to RNA is called transcription, and then RNA to protein is called translation. And so, as I said earlier, this is the central dogma, where we get from DNA to mRNA to protein. And so that is the basic process that occurs in our body every second that we're living, and that helps us stay alive and helps our body function properly.
And now on to going more in depth about transcription and translation. I will be talking about transcription, and Kishan will be talking more about translation. And so now for transcription. Well, what is transcription? Transcription is honestly the process of copying DNA into a single-stranded mRNA. And what this mRNA is, it's a messenger RNA, and that pretty much signals a ribosome to create a certain chain of amino acids. But Kishan will go more in depth on that. And so what happens during transcription is, like I was talking about earlier, your DNA double helix will untwist and separate. And now instead of a DNA polymerase attaching to the strand to create another matching strand, we are having an RNA polymerase attached to a promoter region of DNA, which is kind of like a signal sequence for the RNA polymerase to attach to. And so when RNA polymerase attaches to your DNA strand, you will now make a matching complementary strand of RNA. And what really differs RNA from DNA? Well, in RNA, instead of having a thymine base pair, that T base pair, it's now replaced with uracil, which is U. And what uracil is, it's pretty much just like another puzzle piece that can fit with adenine or A. And that's how you can differentiate RNA versus DNA. And the result of transcription is now you have this, once RNA is processed, you will result in a messenger RNA, and it pretty much carries the genetic message from the nucleus to the ribosome to prepare for translation. And as you can see in this image, As you can see in this image, you notice that there is an RNA polymerase attached to the DNA strand. You have a template strand, non-template strand, and as the RNA polymerase traverses through each single base pair, it is creating a complementary base pair to the DNA strand. And now moving on to a little concept check for you guys. Again, how do the base pairs in RNA pair with each other? And so we'll give you guys 10 seconds to think about that and then review this concept question. And so for the answer, again, now instead of thymine pairing with adenine, you now have uracil pairing with adenine. And guanine and cytosine are unchanged. So A pairs with U and G pairs with C. And now to move on to translation, which Kishan will cover.
Yes, so translation is the second part of the central dogma, where we're converting from the RNA, which is coming from the nucleus, as Max said, to now protein. And so how does that happen? Well, it happens through a process called translation. And basically, we're using a molecule called the tRNA, which is shown in blue on the figure on the right, to translate mRNA, which is coming out of the nucleus, into a chain of amino acids. And so what happens is the mRNA comes out of the nucleus and it goes to a ribosome, which could be located on an organelle in the cell or just in the cytoplasm. And so the mRNA binds to the ribosome. And what you have happen is there's different binding sites on the ribosome to which the mRNA strand is binding to. And so as it moves through, it's basically feeding through the ribosome. As the mRNA feeds through the ribosome, the molecule called tRNA, which is called a transfer RNA, has an amino acid attached to it for a specific sequence of nucleotides. And so this specific sequence of nucleotides is called a codon, which is on the mRNA. And so each codon on the mRNA consists of 3 consecutive base pairs. And these 3 consecutive base pairs match up with something on the tRNA called an anticodon, which is the complementary sequence containing uracil instead of thymine. So still true to RNA's genetic code. And so as it says on the slide, codons and anticodons are composed of 3 nucleotide sequences, 3 nucleotide sequences that match together. And so as you can see, we have the tRNAs coming in and they're complementing the codons on the mRNA. And each time one attaches to a codon, it adds an amino acid to the chain. And that chain is forming at the top of the ribosome, which is shown in green. And so this is called the polypeptide chain. So each time the tRNA comes and binds to a group of 3 base pairs on the mRNA or a codon, it adds 1 amino acid. And so the chain just keeps on growing and growing as the mRNA feeds through the ribosome, and it gets longer and longer and longer until it becomes a super large polypeptide, which is then released from the ribosome when a stop codon is initiated, and it turns into a protein. And so this is the main process of translation and how mRNA turns into a protein via the use of a ribosome and tRNA. And so let's go over one quick concept check. So the question is, how many tRNAs are available in translation? So even though you guys may not know the exact answer, try to think through the concepts that I just talked about in the last slide and that Max talked about transcription, and try to give this question a shot. We'll give you guys 10 seconds and then we'll go over it. All right. So the answer is there's actually 20 unique tRNAs for each of the 20 amino acids that build up protein. So if you guys recall from earlier in the presentation, we said there's a total of 20 amino acids that exist, and there is an actual tRNA for each one of those. So for example, if a certain codon comes in on the mRNA in the ribosome, a unique tRNA for that specific amino acid will come and bind. And there's 20 of those. So you make these proteins with a different combination each time of the 20 amino acids.
So thank you guys for listening in on our presentation about protein synthesis. And we're going to finally conclude this off with a little activity worksheet, which will be attached with this video as well as the answer key. And so pretty much the basic premise of this activity worksheet is you want to, we have provided you with a DNA sequence and have split them up into three consecutive nucleotides, just for simplification purposes. And what we want you to do is to write the corresponding mRNA strand, as you can see in like one of the examples provided. And then based off of that mRNA strand that you have written down, you will label down each of the amino acids that correspond with those 3 nucleotide sequences. And then for the final message, we have for you, just like a little fun thing we added, is each beginning letter of the amino acid, write those down, and it'll create a nice fun message for you guys. And on the next slide, we have provided you with some helpful diagrams to aid you in the completion of this activity.
Yeah, just a quick overview of the left-hand side diagram. As you guys come across different codons in the mRNA, you guys will, for example, if you read GCG codon, you guys can go ahead and take a look on this diagram and start with your G, then we move to C, and then we go to G, and we have the amino acid alanine. And so that's how this chart is helpful in converting from the mRNA codon to the actual amino acid that pairs with it.
And so finally, Kishan and I want to thank you guys for taking the time out of your day to listen in on this presentation about protein synthesis.
Yeah, thank you all for listening, and leave a comment and a like on this video, and go ahead and visit our website at www.guidingpathways.org and check out our Instagram page at pathways_edu. We hope to see you in the next video.
Thank you guys.